Hi, my name is Fabian Tobar-Tosse. I'm professor at the Universidad Javeriana Cali in Cali, Colombia. I will present this poster about the study of bioactive compounds. Specifically, we are working in the definition of key genes in response to bioactive compounds. We are working in the definition of configuration in the promoter region of those genes with the idea to identify possible mechanisms of regulation that support new pathways in response to bioactive compounds. Well, for understanding this poster, it is important to define which are bioactives. These are compounds from plants or from animals that in our cells have the capacity to modulate metabolic process, or in some cases by the interaction with transcription factors. Some of those have the potential to enhance the expression of genes, and in consequence, the industry have the interest in the identification of several bioactive compounds, the sources, and the genes, proteins, and pathways that our cells modulate in response to these kind of compounds. In this study, we analyze the expression levels of genes in response to bioactive compounds. We use the information deposited in a RxPET database. We analyze several experiments for around 20 bioactive compounds, and the idea was the identification of the high expressed genes and which were the bioactive compounds with a high activity at the level of the expression of the genes. Here in this heat map, we are seeing, for example, the high expressed genes for several bioactive compounds, and the most significant result here is that some genes are common for several bioactives. That means that when we apply functional enrichment, it's possible to identify that there is common pathways in response to those bioactive compounds. Of course, this is a standard procedure to analyze of those genes, but we analyze the promoter region of those genes with the idea to identify singularities in the organization of regulatory elements. In fact, here are you seeing a network which connect pathways, but these lines represent the patterns of regulatory elements that we found in the promoter regions. And mainly we found repetitive elements like transposon or simple repeats that allow us to classify those genes based on the possible mechanisms of regulation based on those elements and their binding sites. And we found, for example, that in fact many of these genes and their related pathways are related with the response to signaling, and therefore bioactives could induce modulation in some of those pathways. Now we are analyzing which are the specific pathways and the specific genes in wet lab with the idea to validate according to the configuration of elements in the promoter of the genes. It's possible to recognize essential genes in response to bioactive compounds. For example, here the high and low expression genes, and we found, for example, that the high expressed genes are more confident in respect to the levels of expression. It allows us to validate the fact that there is common pathways in response to bioactive compounds, and now we are working in the definition of which are the specific genes and the possible connections in the definition of new pathways that allow us to propose a more clear pathway or common pathway in response to several bioactive compounds. And now we are working in this kind of analysis. Well, this project is funded by our Ministry of Science and Technology, MinCiencias, and if you have any question, please, this is my email for future communications. Thanks for your attention.